So how's everybody doing? Tired? Me too. I'm like exhausted at this time, but it's okay, one day at a time, right? And I know there's a lot of information in these lectures, right? And immunology doesn't make it any easier, right? So let's really try to get down the things that are more important, right? So we're going to continue where we had stopped last time, and I cut some of the less slides out of this lecture because I think like 43 slides is plenty. Is the mic working? Can you guys hear me in the back? Everything's cool? Yeah? So before I go on, do we have questions about what we covered so far? Yeah.
So what are the main differences between B and T cells?
Oh, that's a big question. Okay, I'll mention some of it. Okay, question is, what's the difference between the B and T cell development? Well, that's a very, very big question. All right, so in order to make that information sink in a little bit more, and also because I had a student who was a bit confused about what this chart means, so I added here again, just to make sure that everybody knows what's going on, right? So yes, okay, so what is happening here? For the B cell development, right? So we have 4 main stages, right? And what is this chart here doing? What is it telling you? Well, it's telling you that, first of all, everything is happening here in the bone marrow, right? So your B cell is developing in the bone marrow. And this is telling you what's happening across the time, the whole development of this B cell. So this chart here is telling you which genes are, or which process is taking place at the stage of that cell being developed. Okay, so what happens here? In the stem cell stage, this cell can become anything that it wants to, right? But once it receives certain signals that are coming from the stromal cells and endothelial cells that are in the bone marrow, do you remember what those signals are? There is one interleukin.
Interleukin 7.
Yeah. You want to c-KIT, interleukin 7? Well, yeah. And c-KIT, what's that c-KIT? I need extra chocolate today. Yeah, here. Okay. Yeah, I definitely need chocolate today myself. So anyway, I can't really talk and eat chocolate at the same time. So here you had the c-KIT and you had interleukin 7, right? And you can develop your own mnemonics of how to remember that. But you can think that this stem cell is so lucky that it's going to become a lymphocyte, right? So you give interleukin 7, right? And how you remember c-KIT? Well, I don't know. Just think about a cute kitten. So a cute and lucky kitten. There you go. So now you have your lymphocytes. You have your lymphoid lineage. All right. There are other signals, of course, that are going to tell this almost lymphocyte to become a lymphocyte and then to become a B cell. But I don't want to put too many of those weird acronyms here because it's just going to make your brain a bit more confused than mine too. Okay, so we stick to the simple, right? So this cell in the bone marrow received those initial signals. It's going to become a B cell. The first thing that it's going to do is that it is going to divide. It's going to proliferate a lot. So this is in the beginning stage before it becomes a pro-B cell. Okay, so first thing it is doing is proliferation. So this is why you have this bar that shows up here, because that's the time, right in the beginning, that this cell decided to become a B cell and is going to divide a lot. It wants to make a lot of daughter cells because it doesn't know how successful it's going to be at rearranging the B cell receptor. So the more it tries, the more the probability that it is going to get a right B cell receptor. So it divides a lot, divides and divides and divides. Is anything else happening here when the cell is dividing? Well, that's when you should look at the chart and see if any of these other genes are on, and they are not. You don't see on bars here. So this thing is on, it's dividing, nothing else is on, right? And you should know why that's the case, because those very important genes that are going to go on after the cell stops dividing, which is right there, right? Those genes that are going to go on, they are going to start messing up with the DNA of the cell, right? Or the DNA of the somatic cell. That thing is going to start to recombine things, right? So you have a recombinase there. And besides that, you have this other enzyme here that is going to start adding nucleotides without a template, okay? Remember that when you do cell division, you're always doing semi-conservative replication. You always have a template. Now, when you start doing this recombination and this addition of nucleotides, this thing is happening without a template. It's just adding nucleotides because it can, okay? So first thing that is going to happen here is still you look at this B cell, you know it's B cell because there are certain markers that you can identify it as B cell, those are not showing here. The thing that is happening here is all inside of the nucleus. You start doing this rearrangement inside of the nucleus, but nothing here is at the cell surface. So this first stage here, you call it your pro-B stage. So the cell is bald, nothing on the cell surface, but things are happening in the nucleus. DNA is starting to get rearranged and nucleotides are being added. So all this stuff is happening inside of the nucleus, okay? So then what's going to happen? Once you make, which chain you make first? Yes, we said that. Yeah, get on track. Right? Good catch. Sorry, I almost hit. So you have your heavy chain first, right? You remember also the sequence of how you're going to rearrange those genes. Which ones are the two genes that are going to get pulled together first?
DJ.
DJ. You got to remember that, right? Cell got to stay alive. Okay, DJ. After DJ, you're going to get VDJ. Now, that part of your heavy chain, that's the only part that's going to be rearranged and mutated somehow, okay? That's the variable region of your heavy chain. When you're done with the variable region recombination, you're going to add your constant region to it. The constant region is the part that is going to be after your variable region and is going to attach that part to the membrane, okay, to the cell membrane. That's going to be part of your B cell receptor. The constant region does not get rearranged. It's always constant. That's why you call it constant, okay? The other thing that gets rearranged, that's your variable. All right. So here you made your heavy chain, and remember you have to test your heavy chain to see if it works or not. And today we're going to see a little bit more detail on how you do that testing, okay? So here, when you have only the heavy chain on the surface and you have to test it, the rule of these B cell receptors is that you're not going to test the heavy chain if you don't have the light chain. But of course, you're not going to make the light chain if your heavy chain doesn't work. This is when you bring in that fake light chain. And what do you call that? Surrogate light chain, right? That's your surrogate light chain. So you get your surrogate light chain over there, and if it interacts with your heavy chain, that tells the cell that your heavy chain is functional, okay? This is happening at your pre-B stage. So pre-B, you have heavy chain, you have your fake light chain, right? If it is functional, you are going to be really happy about it. And what happens when you're really happy? Not yet. Not yet. You procreate. In this case, the cell divides. Yeah, okay, just remember that. Okay, so you get really, really happy. Your heavy chain worked. So you're going to stop those genes that are doing all this rearrangement, and you're going to start dividing again. You're going to divide a lot. This stage right here, between the pro-B and the immature, is the stage in which your developing B cell replicates the most, right? It replicates the most at that stage. because it already went through a lot of energy and effort to make the heavy chain, to make it work. Okay, so after it checked that it worked, it divides. Because what's going to happen? Every one of those daughter cells, and there are many of them, every one of them is going to have the chance to make a light chain, and every light chain is going to be different than the sister's light chain, because each one of those cells is going to have its own rearrangement going on. Okay, so you're going to have daughter cells that have the same heavy chain but different light chains. Okay, all right, so okay guys, let's pay attention here, because you know, I can't quite function with a background noise. All right, so here your heavy chain worked, procreated a lot, they replicated a lot, right? Then you're going to start making your light chain. But if your heavy chain did not work, what's going to happen to the cell? Not yet. You can start thinking about killing it, but it has one other chance. It's a heavy chain, right? This is a heavy chain. It's going to use the other chromosome, because until now, both chromosomes started to make the heavy chain, but one got put on the surface first and it got tested first. Yeah. Yes, allelic exclusion, if it worked. If it did not work, then that chromosome gets shut off, gets shut down, and the other chromosome gives it a go. Then the other chromosome tries to make the functional heavy chain. If that one works, very good. If that one did not work, then the cell dies. Okay, so the cell has two chances to make a functional heavy chain, right? So now let's say it works, one of them worked, and remember that allelic exclusion, because that is a very important principle, okay? So now it worked, now you replicated a lot, now you're going to be ready to upregulate your recombination genes again, because you have to recombine your light chain, okay? Remember, for the light chain, you're going to have 4 chances of getting it right. And usually, for the light chain, the process is not as chaotic as it is for the heavy chain. You have better chances, mechanistically speaking, to get the light chain right. Okay, so first you're going to choose one chromosome from one of your parents that has a light chain. So you're going to inherit 2 chromosomes from each parent that they have one type of light chain in the genes, right? So the first one that is going to be used here is going to be the kappa of either parent, or it's going to be the kappa light chain. If that one works, then allelic exclusion, nothing else needs to work. If that one fails, you try the other kappa, so that's your other parent. If that one fails, then you try, which one? The lambda. And if that one fails, you try the other lambda. And there is something else that I didn't mention, but we'll get to it later, is that if that one fails, you assume the cell is going to die. But no, they make it more complicated. Okay, B cells, after all of this rearrangement failed, they have a way to modify the variable region of the receptor to try to make it work again. Okay, just keep that in the back of your mind. We're not getting into that right now. We're going to get into it later. Okay, but you can still try to salvage the cell. Okay, all right, so now let's say you upregulated a RAG1, RAG2 genes. Remember, you do not have TDT and non-template addition for the light chain. That's only for the heavy chain, right? So now you make your light chain, and it works. It tested, it bound to your heavy chain, and now you have to do something else that is very, very important. What is the next step? Negative selection, exactly. Now you're going to test if the receptor is going to recognize itself or not. You don't want it to recognize self, otherwise you're going to have autoimmune disease, autoantibodies, and those are bad, okay? So you're going to test, and all this is happening in the bone marrow. So the repertoire of proteins and things that you are testing against is not as vast as what is happening in the thymus for the T cells. Okay, so different things going on here. Remember, in the thymus, you have the very special transcription factor that is called ARI, right? Air, moving there. ARI transcription factor, okay? And that one is going to make those pieces of protein, right? It's going to be put in MHC proteins, right? Presenting platters, okay? But here, it's not happening. This with the B cells is going to recognize something that is like 3D, right? It sees things in 3D, okay? So it's going to go through the negative selection. And if it passes negative selection, if it survives, then this cell is going to send a signal to the nucleus to do some strange things with the transcript of the RNA that is making the heavy chain. Okay? Remember the guy, the heavy chain? The heavy chain is what's going to tell the immunoglobulin what class it is. Okay? So the first class that the B cells make, it's always IgM, because the mu gene for the constant region is the first one that comes after all that variable stuff. Okay, so that's always the first one, it's always IgM. Okay? Once you test it and it passes negative selection, now the cell sends a signal and says, you know what, start to do some processing of the RNA that will give you different proteins that are coming from the same RNA. Okay? So in this case here, you are going to have one RNA molecule, and you're going to do differential splicing, alternative splicing, right? The proper name for it is alternative splicing. And you're going to end up either with IgM or with IgD. Okay? Those two are going to go to the surface of the cell, right? Remember I said this cell here also is going to have IgD right there. So this guy here is going to be IgD. Once you express IgD on the surface, that is a signal for this cell telling it that it has matured successfully and it can leave the bone marrow and it can go into the periphery. And what is the periphery? Everywhere else in the body, but not in the bone marrow, basically. Okay, so the cell is going to navigate out of there and it's going to explore the rest of the body. And then it's going to circulate in your blood, it's going to go to the lymph, it's going to go through the lymphatics, then it's going to go back in the blood, just because it likes to travel. Okay, it likes to go around the body. Not as much as the T cells. T cells like to travel more. Okay, we're going to see today. Okay, so that's the story with your development of the B cell. Okay, so this is time that is going across this way, and those are the processes that are happening depending on which stage, and those are the genes that are getting upregulated depending on which stage. Okay. Does that look a lot more clear now? I would hope. Yeah. Okay. So you do the same comparison when you look at your T cell development, because they follow some things that are similar and they have some other things that are different. Did I put T cell here? No, I didn't. One of the major differences between the B cell and the T cell development is that the T cell has another checkpoint that is going to check for recognition of those MHC molecules, right? So that is your positive selection, okay? That happens before the negative selection for the T cells. So make sure you understand that process for the T cells. All right. So what is happening here? I put this slide here again because I don't think I emphasized enough what's going on here. Okay, so what is happening here? Look, this is inside the B cell DNA. What DNA? Well, DNA for your heavy chain. Okay, your heavy chain has this area that was first, it was recombined, right? When you had all this variable region recombination, you have lost some DNA because it got cut out. You just lost it. Okay, you pick some DNA that come in there, or the DNA, you get rid of it. Okay, so you end up with this piece that is VDJ, because this guy is the heavy chain. He has the D, right? VDJ. Now, when you look here at the end of this gene, you are going to see the constant region genes. So every one of these little rectangles here, that is a constant region gene. This is a gene, that's a gene, that's a gene, okay? Now, when you look at these genes, you're going to see that each one of those here have a little green dot in front of it. That's the dot, that's the dot, that's the dot, that's the dot. There's no dot here. There is no dot there. That dot is called a class switch sequence. So I put it right here. Okay. It's called a class switch sequence. The only gene that does not have a class switch sequence is the delta gene. Okay? What it means is that when this gene, when this whole thing gets transcribed from DNA to RNA, to messenger RNA, right? Remember transcription, right? When this thing gets transcribed, you are going to transcribe, first you're going to transcribe the whole thing, but then when this part here gets transcribed, they go as a unit, because those things, not only the delta does not have a class switch, but it has something else. Okay, so let's put here, this is like the end of your RNA here. So here you're going to have your class switch, okay, class switch right there. So here you're going to have your mu, and then after that here you're going to have a poly A sequence, okay, and then you're going to have here your delta, and after the delta you're going to have a poly A sequence. Poly A sequence. Okay, this is your mRNA, so it's going to have to be processed. So after your light chain was found to be functional, that is going to send a signal to the nucleus to tell this RNA that it can undergo alternative splicing of the messenger RNA. So this is all happening inside the nucleus. Okay, so after you test the light chain works. Now this RNA has the option. Either it's going to stop right here, or it is going to cut this piece out by splicing out the exon, right? So this is an exon that gets spliced out, and then you're gonna, instead of keeping that part, you're gonna keep the part that has this thing with the poly A in it. Okay, so that's your alternative splicing. And this is how, in the mature B cell that is still in the bone marrow, you are going to end up with two types of antibodies on the surface of that cell. Now, notice that the variable region is exactly the same. Nothing happened to that. It's still there. It's going to be right here. The variable region is going to be right there. And it is exactly the same for the IgM as it is for the IgD. The only thing that's happening here is a swapping of the tail. So the tail, which is in the heavy chain, is going to tell the isotype. If it did not do that alternative splicing, it's going to be M isotype. If it does the alternative splicing, it's going to be D isotype. So both of them are expressed. When D is expressed, that is going to tell the cell that it can come out of the bone marrow and go travel in the periphery. Okay, is everybody cool with that? All right, so those green things that are the class switch sequences, okay, what is the use of them? Well, for now, nothing. Those things are not going to be important until after the B cell finds the antigen that it was made for. Okay, so nothing is going to happen here if it doesn't find the antigen. Not only it has to find the antigen, but it needs to be activated by a T cell. Without the T cell activation, the B cell will not be able to class switch. Okay, so B cells, they have the capacity of identifying their antigen, their target stuff, and they can produce antibodies, but they are going to only be able to produce soluble form of IgM antibody if it does not have help from the T cell. Okay, so no class switching if you do not have a T cell. All right, so is everybody cool with that? All right, yeah.
Do B cells undergo positive selection?
No, what is needed to undergo positive selection? MHC. B cells don't need MHC. They see things in 3D. They don't need to see the little stretch of protein in a platter. That's the job of the T cell. T cell only sees 2D and only if it is presented in the platter. So T cells have to undergo positive selection because you have to make sure that the T cell is binding the right way to the MHC. Okay, so that's your positive part of it. Okay, everybody cool with that? Yeah, okay. All right, so now we're going to the next parts here. So let's see what happens here. A little bit more on that N nucleotide addition, okay, because I didn't mention this much before. This is only happening for the heavy chain, okay? So N is for non-template. So this thing is just adding nucleotides out of thin air just because it can, okay? Non-template, add the nucleotides, and you have an enzyme for that. The name of the enzyme, thank goodness, is terminal deoxynucleotidyl transferase, because it goes to the end of that gene over there and it just adds nucleotides, and hopefully it's going to be part of a codon and it's not going to bring the thing out of frame. Okay, so adding non-templated nucleotides over there. Okay. Is that clear? You can see the mechanism in your head? Hopefully? Okay, I'm assuming that everything is clear. All right. Okay, so clarification here for your large pre-B stage, okay, when you have your surrogate chain that is testing your heavy chain. Okay, so picture here, you're testing your heavy chain. What's going on here? Remember, your heavy chain is always going to be the M, heavy chain is coming from the mu, because there is no such thing as class switching here. Class switch is not going to happen until much later, right? So this is always your M antibody type, is your M immunoglobulin, okay? Now look at what happens here. The tail of that M heavy chain is very short. So when you are testing the heavy chain, you really want to know, can it send a signal to the inside of the nucleus or not? But the heavy chain itself is not capable of doing anything because it doesn't have the tail for it. There is nothing here in this tail that can send a signal to do signal transduction to the nucleus. Okay, so what happens here is that once you produce your surrogate chain, remember those pieces of 2 proteins of your surrogate chain, you have something that is kind of variable, right? Is not variable, but is mimicking the variable binding site. And then you have the other one that is the lambda site. So your pre-B protein here is made up of lambda 5 and variable protein, okay? And V pre-B, okay? V pre-B protein, okay? So these 2 proteins are interacting with your heavy chain. And what happens here is that the interaction, which is not showing here, the interaction of your surrogate chain is going to allow the proximity of these other proteins that are also on the surface. That is your Ig alpha and your Ig beta. Now these proteins here have a long tail, okay? So once these chains are interacting successfully, that is going to send a signal to these guys over here to start a process that is going to start a signal transduction, okay? So these Ig alpha and Ig beta are very crucial for sending this signal that you have here, what you call ITAMs, that are going to put some phosphorylation and change some modification on proteins that are going to send a signal that this cell can go ahead to the next stage, okay? So what happens? Once the signal is done, then you are going to stop, let me see. Okay, you're going to stop those RAG1 and RAG2 as well as the TDT genes from being expressed, and you're going to start proliferating. Okay, so after you check your heavy chain, that is the stage that gives you the most expansion of that clone. What clone? The clone of your heavy chain. You just made a clone. Okay, so that's the time of largest expansion of the cell. Okay. So here is the story with the Ig alpha and the Ig beta. Okay, so they're very close, and they are part of your B cell receptor. Okay, the one that has the surrogate chain that is called your pre-B cell receptor. Now, once you have your light chain, then you call it the B cell receptor. So the B cell receptor is not made only of your immunoglobulin here, of your Ig thing here, IgM, but it's also including those Ig alpha and Ig beta proteins. Okay, so this whole thing here is your B cell receptor. Okay, so what happens to this thing here? Because it has a long tail, okay, it has this very important places that they have these tyrosine activation motifs. Okay, so the name of these domains is actually immunoreceptor tyrosine based activation motifs. You call them ITAMs. Okay, so ITAMs inside of the cell, inside the cytoplasm, they are going to get the signal when your IgM detects antigen, when it interacts with antigen. If it's not interacting with any antigen, then there is no signal to be sent to these guys over there. Okay, and this is, of course, for your regular B cell receptor. When you look at your pre-B cell receptor, the job of your surrogate chain is to pretend that there is antigen so you can test this whole thing if it works or not. Okay? You call it a gamma globulinemia. At least recognize the anemia part of it, because you can relate to when you don't have like red blood cells, you call it anemia, right? Now you look at this and say a gamma without gamma globulins anemia. So without gamma globulins anemia type. That's the type of the disease. And lack of this Bruton kinase guy is one of the most frequent types of X-linked. That means Bruton is in your X chromosome. Remember your chromosomes? Which one is the X chromosome? Is the one you get from your mom. Okay? So your X chromosome, well, if you're a girl, you could also get it from your dad. But if you're a guy, you get it from your mom, for sure. Okay? Now, the X chromosome linked, I mean, the Bruton is on the X chromosome. So who do you expect to have more problems with this disease in the general population? Males, right? Because the males inherit only one X chromosome and the other one is the Y, right? So females can be heterozygous, but they'll survive if one of the genes is working, right? But if the guys inherit the bad X, they are in trouble. Okay, so this is the X-linked gamma globulinemia. Okay. Failure for B cell maturation means you don't get B cells. All right. Now we come to the question, which I also just gave you the answer. That's too easy, right? Yeah. Once you have recombination of your heavy chain, that's the time you start to make those proteins as well. And those are not recombined. They're just coming from the genes. So they're going to be expressed at a time. Yes, yes, because you have to test your heavy chain by having those present too. So they have to be expressed before testing. No. Okay, so what is your answer? It is B. You're going to have a lot of problems with bacterial infections because you don't have B cells to eat the bacteria. You don't have antibodies. Okay? Now, I had a few people who answered A. You're not going to have excessive B cell proliferation because they're all going to die in the bone marrow. You're going to say you have excessive B cell death, like all of them. Okay. All right, so here, checkpoints for B cell maturation. The reason why I did not put the source of these slides is because there are some points here that are not correct in the source, so I just deleted it and I'll just use the points that are correct, okay? So what's going on here? Look, this is all happening in the bone marrow, okay? So first, you're gonna have your commitment to become a B cell, okay? Remember I mentioned before interleukin 7, I remember c-KIT, but then there are other factors that are, transcription factors that are really going to commit the B cell to be a B cell, and those are right here, E2A and EBF. So those are, you can say, like expression of the B cell best friend. Okay, there are other best friend things here for the B cell, but this one, it really has a E best friend that is going to tell it to become a B cell. Okay, so now it's committed to be a B cell. Then here, after you have your pre-B cell receptor, remember what is making the pre-B cell? Is your heavy chain with the surrogate alpha chain and your Ig alpha, Ig beta. That's your pre-B receptor, okay? So here is going to be the first checkpoint for survival. Testing of the heavy chain is the first checkpoint for survival. That does not depend on anything binding to the variable region. This is why it's called antigen-independent checkpoint. Quite simple. Okay. Then what happens? Then you're going to make your light chain, heavy chain works, blah, blah, blah. You go pre-B, blah, blah. You get your light chain over there. Then you're going to have to test whether your B cell receptor recognizes self or not. So this is your second checkpoint. That is your negative selection. Okay? This is happening in the bone marrow. But this is antigen dependent. You have to recognize something here in order to pass the test. Okay, so two checkpoints in the bone marrow. One that is antigen independent and one that is antigen dependent. Okay? And this is when something gets interesting, because at this point, on your second checkpoint, if you have a B cell that reacts with self, the B cell tries to save itself, and it goes through this light chain receptor editing process. This I had not mentioned before. So what is this cell going to do? It's going to say, my receptor looks too good for me to die. I'm going to try to rearrange my variable regions again. So it's going to upregulate the RAG genes. And it's going to try to get some other genes to modify the variable region. And it's going to test it again. Because the rest of the antibody works fine, interacts fine. Okay, you have the heavy chain that is interacting with the alpha beta. You have the constant part of your light chain that is interacting with the heavy chain. Everything is fine. The only problem here is the variable region. Those are giving problems. So this B cell is going to say, oh, let's try this trick. Upregulate the RAG genes and let's modify the variable region and let's test it again. Sometimes it works. Sometimes it doesn't work. And the cell is going to try to test it based on how many genes it has left to substitute there with the RAG 1 and 2. When it exhausts all its possibilities, then it is going to give up and die. Okay? But it's trying really hard to salvage all this effort that it put to get to that stage. Okay, so this only happens to the B cells, not to the T cells. Okay, only B cells. So this is another difference in that. Okay. Okay. Now, once you pass that stage, right? Once you pass the second checkpoint, then you're going to have the third checkpoint that you're going to be, of course, you remove your self-reactive clones, you kill them, right? And you are going to do that alternative splicing and allow the cell to go to the periphery and circle around until it finds the antigen. Okay, so proper alternative splicing here has to do with another checkpoint. Okay, so no, there's another checkpoint after this. When we're talking only about the bone marrow, these two checkpoints. This checkpoint here for negative selection, recognizing self, that is called central tolerance. That is happening inside the bone marrow. So if it is in the bone marrow, it's central to where the thing is developing. It's called central tolerance for the B cell. Where is the central tolerance for your T cell? In the thymus. Okay, so that's central tolerance. Once this cell goes to the periphery, you're going to have another checkpoint that is going to be peripheral tolerance, which we'll get to some other time. Okay? So you have several checkpoints because you want to make sure that you're not going to have autoimmune disease. Okay? All right. So we talk here about alternative splicing. Yeah, and that is RNA processing. It happens inside of the nucleus. But if your RNA is not processed properly, all messenger RNAs go through this process to RNA processing. Some of them undergo self-splicing, some other ones need spliceosomes, right? And then you have, first thing that happens, of course, you put the 5' cap. Remember that thing with the 5' cap? And then, you go through the splicing, and then you put a poly-A tail, right? And if the RNA is processed properly, then it can be exported to the cytoplasm, right? Goes through the nuclear pore complex. I like the nuclear pore complex, the basketball kind of thing. Okay, so that is happening to the RNA. Okay, so we got that. Okay. Now, after all that, when your B cell goes to the periphery and then it finds antigen, it's going to be able to again modify the binding, the variable region. And I'm going to see that next. Okay, and this is what we call somatic hypermutation. This is only going to happen to the B cells and only after it finds antigen. And more specifically, after it finds antigens and it gets a kick from the T cell. Okay? Because if the B cell finds an antigen by itself, it's not very good at doing this somatic hypermutation. Now, what is this thing? When you look at the variable region, after second exposure to the antigen, you are going to tell the cell to expand, to replicate, right, to divide, and to make clones of itself. And every time the cell is dividing, those variable regions, actually there are three parts that are more than variable. They're called hypervariable regions. They are going to be mutating to change the affinity of the antibody to the target antigen. Okay? How does that happen? Well, it happens just by chance. It's not like the B cell is trying really hard to get the best affinity. It's just changing nucleotides and then doing the transcription, translation, and let's see what happens to the protein. Totally random. Okay? Of course, that is only going to happen in the variable region because your constant region is constant. Okay? This is going to happen after you have certain exposure to the antigen. You don't need to make better affinity to antigen if you don't have the antigen, because you've never seen it. So you think you're good enough. After you see the antigen, you say, oh, I can't kind of grasp this guy, and let me change my affinity after antigen. Okay? Okay. Now, this is what happens to your variable region after the cell sees antigen. Not right away, because the cell has to divide, and then of course this is going to happen with the help of your T cells, right? Because B cells by themselves can't do much, okay? So now after sometimes some days that you're exposed to your antigen of binding, okay, like how many days? Oh, this first thing here is happening 7 days or a week after you have seen the antigen, after you have a signal from the T cell that you have to, you know, do something else with your antibodies, right? So these specific regions in your variable region, they're called CDR1, CDR2, and CDR3, okay? And how many variable regions you have here? Well, we have one for your heavy chain, and you have one for your light chain. So you have two variable regions that are going to mutate. Okay, so you have CDR1, CDR2, and CDR3 for your light chain. And then you're going to have here CDR1, CDR2, CDR3 for your heavy chain. 7 days more or less after exposure to the antigen, you're going to start getting mutation on those hypervariable regions. The mutations are showing up like right here. Okay, so what is this? Clone 1, clone 2, clone 3. These are three different cells that have seen the antigen, and they divided from the parent cell, from the mother cell. So the first cell that saw the antigen was right here. So that's your B cell. Then it divided and it gave three different babies here, three different clones. So each one of these clones is going to be a little bit different than the mother cell. Clone number one started to mutate one of those hypervariable regions. You see, the mutation is right here. But see, clone number 1 didn't do anything to the light chain. It's exactly the same as the parent cell. Clone number 2 was lazy. So you're looking for mutations here, and you're going to say, what is day 7? Any mutations here? No, I don't see any black dots here. No mutation, no mutation. So what happens to the DNA here of this clone number 2? Well, it's exactly the same as the mother cell. It didn't mutate anything. It was lazy. Clone number 3, where do you see a mutation in clone number 3? CDR3 of the light chain, right there. But you don't see a lot of mutations. Now you're going to see what happens to the binding affinity of that IgM antibody on the surface of the cell. What happened to the binding affinity to the thing that is binding, to the antigen? So you're going to say, oh, look at this guy here. This is the coefficient of dissociation. Remember chemistry? Yeah? So if you have a very low coefficient of dissociation, what happened to the things that is associated? Huh? It's tighter binding. That's why the face, right? If you have low dissociation, that doesn't want to come out. If you have a high dissociation, dissociates easily. So you look at those numbers for dissociation constant, and you say, which one here binds tightly of these 3 clones of day 7? You have 33.3% chance to get it right. All right, let's wake up. Which one of these 3 clones on day 7, which one is binding tightly to the antigen? Yes, but you got a chocolate already. All right, clone 1. Why? Because you see the number here, and this is the smallest number among them all. Okay, you look at the parent clone. Well, the parent is over there. Well, this guy here did not mutate, so this guy is the parent clone. Your dissociation constant is 4. You look at this other cell, number 3, dissociation constant is 6. That means that mutation made the situation worse. So if this is your parent cell, that kind of like, all right, I'll hold on, but not too tightly. clone 3, he's like, forget it. It doesn't want to know about the thing that is binding. It doesn't want to bind. Okay? Now, let's see what happens 2 weeks later. 2 weeks later, same cells over here, right? Now, look what happens here to these other clones here. Okay, 2 weeks later. This clone number 4, 2 weeks later, he had 1 mutation here, he had 2 mutations there, and this, ooh, look at that dissociation constant. Look at that. That is low. 0.4 is 10 times stronger than the parent cell. So that guy is a good clone. He can stick and hold on to it. Look what happens here after this one's a secondary exposure, right? But basically you can see here, most of these clones here, after 2 weeks of exposure, most of those clones have better affinity for the antigen. And you should be asking, what happened to the ones that had bad affinity? Well, they just didn't bind the antigen strong enough, and they didn't have enough stimulation to stay alive. Because B cells have to be stimulated to stay alive. If they are not stimulated, they get bored and die. So the clones that did not get a better binding, they disappeared. You're not going to have clones that are useless, or they can become zombie cells. They just go around like doing nothing. Not binding, zombie, you know. But you're going to see zombies later. Okay. So day 14, you have stuff that has a lot better affinity. And then if you go and you give a booster shot, expose those cells to the same antigen a second time, look what happened. What tends to happen to the differentiation of this hypermutation, okay? Some of them get worse, but some other clones get a lot stronger binding affinity. So the more times you expose those cells to the same antigen, the better antibodies you can get out of it. And this is one of the reasons why you have to get booster shots for vaccinations, depending on the vaccination. Okay? All right, so everybody cool with that? Somatic hypermutation, that's the name of this game. Does it happen to T cells? No, this is a B cell thing. All right. So, after the B cell sees the antigen, and it has the help of a T cell to do the class switching thing, because remember, it's not going to do it by itself, right? After it binds to CD40 ligand on a T cell, right, then it can decide to change the antibody isotype. Okay? So the first antibody you're going to make, no matter if it sees the antigen by itself or not, first antibody you're going to make is IgM. You're going to spit out IgM. If you get the stimulation from the T cell, now you can switch your antibody type. Well, what is the type that you're going to switch next? Exactly, we don't know. We don't know. Is it because you don't understand the question or because you don't know which antibody it is? Well, we don't know. Because it depends on the pathogen that was found in the first place. Okay? So you're going to have a choice of switching the class to either IgG or IgE or IG, which one is missing? A, that's it, right? Because you start with M and D is like, you know, useless kind of to the systemic, right? So you can switch to one of those 3 types, but it really depends on the antigen that you are going to look. But one thing that is important about this class switching is that the cell that does the class switching can never go back to IgM, because the piece of the chromosome that had IgM gets cut out. It gets thrown away. So the DNA of this cell that did the switching is missing a part, is missing the piece that could make IgM. It doesn't have it anymore. Okay? So you see, you can make other classes like IgG, you have IgG1, IgG2, other types of IgGs, but you can never go back to IgM. Okay? You could class switch to something else that is down the line. But the piece of chromosome that is gone, it's gone. Okay, so that's the purpose of this slide here. You see that this class switching here got close to the constant region that is constant epsilon. So the type of antibody that this thing is going to make is going to be IgE. Okay, and IgE is a monomer. Okay. So remember your antibodies. All right, so here, after the B cell is exposed to antigen, it starts to change its personality. So it was kind of peaceful and quiet, and it had all its hair in its head, right? And it was minding its own business. And then it found antibody, and it became an antibody shooting machine. There you go. It starts to shoot out antibodies. Okay? So the first one is going to be your IgM, and then it might class switch or not. Okay? It becomes a plasma cell at this point. All right. Now, your types of B cells, right? You have two main types of B cells. The ones that we're more concerned with is B2. That's the one that is working in the systemic, right? Systemic circulation and in your lymph and everything else. Now, besides your B2 cells, you also have B1 cells. Now, these guys are weird, okay? Because first of all, they are not really developing in the bone marrow. They don't even develop after you're born. They develop in the liver and also in the spleen to a lesser extent, but this is before you're born. So these guys are dividing and developing in the liver of the fetus. By the time you are born, you have all your B1 cells that you're going to have, all the B1 clones that you're going to have, okay? And then you should ask, like, what, these cells are going to last forever? No, but they are self-renewing, okay? So you got them from your liver before you were born, and throughout your life, these cells keep on renewing themselves. Okay, they divide and they make more, clones of B1 cells. Okay, they are very important for mucosal defense. Where do you have mucosa? In your body, in your lungs, and in your gut, and ladies, they're there. Okay, so 3 places, basically, okay? So these B1 cells are very important for defense against stuff that want to go through the mucosal barriers, okay? And these B cells, they are very cool that they are like self-sufficient, okay? They are going to get activated without the help of T cells. So they are T independent. Who needs T cells anyway, right? And they produce IgM antibodies, but they also produce IgD antibodies, okay? And those are important for mucosal defense, okay? So that's the weird thing about these B1 cells, right? They are able to produce antibodies without help of T cells, and on top of all, they can even produce antibodies without any stimulation from antigens at all. They are totally self-sufficient. Antigen, who needs them? I'll just produce my own thing. That's the B1 cell, okay? We're not going to focus too much on them. What we focus on is really your B2 cells. But these guys here are even strange to study, right? You don't even have proper animal models to study this thing. Because animals have different immune systems, and what you're going to do is study humans? Try to get an IRB for that. I'm done with research. Okay, so B cells, right? Your B2 cells, that's the one we're talking about, okay? And we already talked about that. They produce high affinity antibodies after class switching, after seeing antigen. All right, so that's the story with the B cells so far. Is everybody okay with it? Yeah, can we start diving into migration? Migration, all right, so let's see here the migration of lymphocytes, right? What is happening here? You have dendritic cells, right? That are sitting in tissues. So those are your interstitial dendritic cells. And they are waiting for some pathogen to come along. Remember those guys, the dendritic cells? Hopefully, yeah. So they are waiting for some pathogen to come along, and then they are very good at eating the pathogen. And remember, at that time, they are phagocytic, and then they're going to move to the lymph nodes. And I mean, first they're going to become non-phagocytic, right? They ate enough, and they ate so much, they got all this energy, they're going to run somewhere. So they're going to go to the nearest lymph node, carrying that pathogen that they just ate. Okay? How do they get to the lymph node? That's the story of migration. Okay? So they are going to go to the lymph node and enter the lymph node by the vessels. They're called afferent lymphatics. So imagine that this square thing right here is a lymph node. Okay, in case you didn't see the lymph node there, I hadn't seen it first either. Okay, so that thing is a lymph node. Now, your lymphocyte, your dendritic cell can get there in two different ways. If it gets a pathogen that is out in this interstitial fluid, so is out of the blood vessel, okay, then what it's going to do is going to hop here. It's in the lymph anyway, so it's going to hop in lymphatics and go through the lymphatics and reach the nearest lymph node, because it doesn't want to go too far. And it's, of course, practical for it to reach the nearest lymph node. Now, the name of that vessel that reaches the lymph node is called afferent lymphatic vessel. And you can imagine A for arriving. Okay, so it arrives there. Now, this picture is not very fair to these afferent lymphatics, because you don't really have just one afferent lymphatic. You have actually several afferent lymphatics. Okay, you have several afferent lymphatics. And then in the other side, you're going to have like one efferent lymphatic. Okay, so this thing is going to get there through the afferent lymphatic and is going to look for a T cell that matches the antigen that it just ate. So it's going to sit in the lymphatic, in the lymph node, and it's going to look for a T cell. Okay. Why? Well, because these dendritic cells are very good antigen-presenting cells, and you even call them professional antigen-presenting cells. Okay, so there are some characteristics to these cells that make them professional. Okay, so what is this here? Okay. Okay, they have this co-stimulatory molecule that is called B7. Okay, so this is your dendritic cell. Okay, it has B7 on its surface. Now, it already had some B7, but after it ate the pathogen, bless you, it made even more B7. So this guy is like a B7 production OCD guy. He's going to make a lot of B7 on its surface. Okay. Once it has all this high level of B7 molecules on the surface, that's what makes it such a good antigen-presenting cell. Okay. Besides B7, this cell also has to have that platter to present antigens to your T cell. So it has to have MHC molecules. Okay. Now, if the thing that it ate was some extracellular thing, so this thing is going to present a piece of the pathogen on MHC class 2 molecule. Right? So let's keep that in mind. But it needs MHC molecule. And is also going to start upregulating the MHC molecules on the surface. So this thing is going to be decorated with B7 and with MHC molecules. And it's going to go to the lymph node, and it's going to sit there and wait for the right T cell to show up. Okay? Now, let's see. So in the lymph node, it's going to sit there, And now the T cells are going to get to the lymph node. They're going to transit. They're going to, float around. They're going to get to the lymph node. If the T cell recognizes the antigen, then it's going to start developing. If it doesn't recognize the antigen, the T cell is going to go out and circulate again. If the T cell recognizes the antigen, it's going to get activated by the dendritic cell. And then the activated T cell is going to come out and go into the circulation. Okay, so the whole action here is going to happen in the lymph node. That's the meeting place. Okay, now let's look back here to what is happening at the first stages of innate immune system recognizing the antigen, because that's the first thing that jumps into action, right? So here you have your dendritic cell, and how is it that it's going to recognize the antigen? Okay, somebody who's awake is still in the back. PRR, yeah, so it's right there. Remember those pattern recognition receptors? All right, so dendritic cells are innate cells, and they have those. So here, it eats up phagocytic PRR, eats up, processes the pathogen, right? Processes the antigen, and it puts it up. In this case here is an MHC class 2. Piece of the pathogen is right there. See the red thing? It came from the pathogen, from here. It's right there now. Okay? Now look what happened. It has MHC. It had already before, but it makes more MHC, and it's going to make a lot of this guy here, that B7 guy. Now, I am very sorry, but B7, just because immunologists are so mean, B7 is also called CD80 and CD86. Okay, but we just want to call it B7. Okay, but this is the other name of it. Not only it had to be called CD, but it had to be called two of them. Okay, B7. All right. So, what's going to happen now? Look at this. This guy here is going to go to the lymph node and is going to look for the matching T cell that can recognize that piece of pathogen. So a lot of T cells are going to come here and test it out, and they don't care, and they are going to just float away. But the one guy that shows up there and recognizes that piece of pathogen, that guy is going to get a signal to divide and to make clones of itself. So this is called clonal expansion. Okay, so it's missing there. You call this part clonal expansion. It's expanding. Okay, the other guys did not expand. Now, before expanding, this thing here was naive. Let me put a division here. Okay, this is like, okay, nothing here. Okay, so before expanding, this thing here was naive. It had not seen antigen. When it sees the antigen on this tentacle of that dendritic cell, now it gets activated. Once it gets activated, this is when it's going to expand. Now, among those expanded cells, some of them are going to become effector T cells. They're going to get out there and do the job. Some other cells are going to differentiate and become memory T cells. They're going to stay with you forever, or nearly forever. They have very long lives, usually forever. Because remember what is happening to your thymus as you age. What's happening to the thymus?
Shrinks.
You get involution of the thymus and becomes fat. And that fat is useless for developing new T cells. So the memory T cells that you're developing, they better be good and for your whole life. Okay? All right. So let me see if I have more questions here. Okay. All right. This was it. Okay, so this is from Cubby saying exactly what I just mentioned. And here another question just before we break. So here is, which of these does not describe a naive T cell? You actually have to think for that. Seconds. The question is which one does not describe the naive T cell? Okay, anybody need more time? Okay, what's the answer? D. The naive T cell is not going to sit there and just wait for something to show up. It's always recirculating. Okay? All right, so we're going to stop here and we'll pick up here from next time.